All right, great. Thanks for everyone for sticking around. I know drinks are coming up, so we'll try to keep this on the lighter side here at the end of the day. But thanks for joining this session on the Innovation Gap, a review of the future of viral vector manufacturing and the delivery of genetic medicines. My name is Alex Stranahan. I cover biotech at Bank of America, and I'm pleased to be co-moderated with.
Hi, I'm Lisa Hensky. I'm a medical oncologist at the Brigham and Women's Hospital, and I do mostly laboratory-based research.
Great, and we have a fantastic panel of industry leaders, mostly representing biotech companies that are addressing several different verticals or strategies when it comes to gene therapy and gene therapy manufacturing. And before we jump into the questions, I'll just say that it's a very timely moment for us to be talking about manufacturing and gene therapy. As an industry, we're moving from a period of treating really underserved orphans, rare diseases with very expensive gene therapies that haven't necessarily required high scale on the manufacturing side for those commercial markets. And given, Parkinson's, wet AMD, DMD, these are significantly larger markets, and the scale necessary to address those markets, even today, is still being developed. So with that, maybe Peter, we can start with you, and then we can open up to the rest of the panel. But in terms of optimizing viral vectors, what are sort of the key parameters in your mind, and what sort of considerations surround this process, including the design of the capsids, and why are they important?
Yeah, well, first of all, thank you for the invitation to be on this panel. It's a great conference. I'm Peter Nasasio. I'm the CEO of Capsida Biotherapeutics. Capsida is a fully integrated gene therapy company with the foundation, really, as the name implies, is capsid engineering. And so a lot of my comments on the panel today will focus around delivery. And so to specifically address your first question, it always starts with what is the problem you're seeking to solve. Using wild-type AAVs as an example are excellent viral vectors for many diseases, like liver disease as an example. It's very easy to get to the liver. But many tissues have a biological barrier that make it a bit more formidable to cross that biological barrier and get to the desired site without getting to non-targeted organs. And so a lot of our focus has been very much on that, specifically in CNS disorders and crossing the blood-brain barrier, and at the same time making sure that we're not accumulating in the liver or in the DRGs. And so we do capsid engineering focused on that task. And so some of the things that are really critical is starting with what's your goal. And what we focus on is creating a target capsid profile, kind of borrowing the concept from drug development of the target product profile, and say, okay, what does the capsid need to look like? What is the brain region that we're most interested in? For an example, we have a program in Parkinson's disease, so getting the substantia nigra is very important. What types of cells do we want to transduce? For us, neuronal tropism is the main focus, but there may be some diseases where you want to focus on astrocytes or glial cells or something else. And so we factor all that into what we're seeking to do up front, and then we use a roboticized high-throughput screening to literally interrogate tens of thousands of tissues. We do all our work in NHPs in order to ensure translatability to humans, and then filtering through those billions of capsids and getting to that one capsid for each target capsid profile that will meet the needs of the disease that we're targeting. And just to say one word about manufacturing, one of the early screens that we do is a manufacturing screen. We have built our own wholly owned on-premises fit-to-purpose manufacturing site that helps us do those assessments very early in the process, so that we know later we're going to be able to have the kind of scale and quality that we need to get to patients and to get to commercialization.
Yeah, I could just ask Peter, are you thinking that there will be one capsid design per disease, or will there'll be some commonalities to where you will end up?
I think it'll be a little bit of both. I think there's commonalities. Many diseases in CNS share common attributes in terms of target capsid profile, being neuronal specific being one of them. But there's certain diseases where you may want to have more spinal cord involvement versus exclusively brain involvement. There may be other diseases where you need to get to other tissues, multi-organ diseases that aren't just CNS focused. As an example, we have a program with CRISPR Therapeutics in Friedreich's ataxia, and that's a multi-organ disease where you want to get to a few different tissue types. But yes, I think for many CNS disorders, having a common capsid could be something that emerges.
Zandy, I know you've done a lot of optimization on the capsid side.
I was just going to mention, I'm Zandy Forbes. I'm the CEO of a genetic medicines company called Mira. We have 4 late-stage, so pivotal phase 3 BLA filing programs. We also do a lot of optimization of our capsid, of our genetic medicines, which includes some capsids, but we tend to use local delivery, not systemic delivery. And one of the things that's really important for what we're going to talk about in costs of goods as well as dose is real optimization of the vector genome. So we optimize promoters, we make the cDNA sequence a little bit more genomic. So when we optimize through transcription as well as translation and mRNA stability, we're talking about improvements in potency of 3 to 4 logs, not 50%. And you can appreciate that if you're improving your potency by that much, you're significantly reducing the dose by 3 to 4 logs that's required for efficacy, which has a 3 to 4 log impact on cost of goods. And it also has a safety impact because the main safety issue we look at, particularly of systemic delivery, is an inflammatory response. And in clinical studies that we've seen so far in the eye or systemically, there tends to be a 1 log difference in the dose that causes inflammation to not. So it's very important when you're broadly delivering to have increased tropism to certain tissues, but in the context of actually making gene therapies for large indications and that are really going to work, optimization of the vector genome as well as the capsid, not just for tropism, but for transduction efficiency and expression is super important. And in the cost of goods arena, just to let you know, one of our programs in phase 3 is Parkinson's, where the dose is so small, the effective dose, and we manufacture ourselves, end-to-end manufacturing and release, that the cost of goods is in the hundreds of dollars, not the hundreds of thousands of dollars. So that's the sort of metrics we're all now working in the industry.
Zandy, you started to talk about the manufacturing aspect of viral delivery, and it's different from many other types of manufacturing, including the regulatory component. Could you talk a little bit about how that factors into your strategy and your goals?
So with respect to manufacturing, we started the company 9 years ago, and there were no good manufacturing facilities. So we built a facility. We now, and that allowed us to do a partnership with Johnson & Johnson. We're now a commercial manufacturer for Johnson & Johnson. We have 2 flexible, scalable viral vector facilities. It's important to understand, people still ask us, what size bioreactors do you have? We don't have a particular size. Regulatory agencies want single use. That means you can swap from 100 liters to 3,000 or 2,000 liters overnight, and your process, our process, is scalable. So you don't have a fixed scale facility. We also have our own plasmid production because our viral vector facilities are GMP and it's under the same quality systems. It's actually GMP. And that's really important because GMP plasmid is hard to find. And in addition, I mentioned we're the commercial manufacturer for Johnson & Johnson. And in order to be able to release something commercially, we had to build our own QC facility because the vendors like BioAlliance, WuXi, all those types of companies are not able to rapidly QC and release material these days. So we have end-to-end manufacturing Importantly, we have a process which is best in class, and we have done more than 20 vectors using that process, over 50 GMP runs. And within, I think, 2 to 3 months for any new vector, we can take that from a plasmid sequence to GMP. This is super important from a value perspective. We save about 3 years on any development timeline. And with respect to regulators, we manufacture everything end to end. And we have filed with 15, 16 different regulatory agencies, and we now cross-reference all of our manufacturing. So we are in a position where, rather than asking questions, the FDA in particular can see our manufacturing and recently said to us, oh, this phase 2 is pivotal because you manufacture. I don't think the agency will lower its standards with respect to manufacturing because viral vector manufacturing is tricky. But I do think that one needs experience manufacturing many different vectors, and in particular, manufacturing for commercial, not for the clinic. And we gained that with a partner, Johnson & Johnson, who's only focused on BLA and commercial, not on what you can get into the clinic.
Hi, everyone. Steve Favaloro. I'm the CEO at Genizin. We're a CDMO that focuses on viral vector manufacturing, from partnering with innovators very early in preclinical to a commercial scale facility we have in Massachusetts that supplies Hemgenix to the market for CSL. And just to build on that, Zandy, it's the specialization in our eyes, too, of having that commercial site, that inspection readiness, and when we partner with late phase customers, the ability to bring them through a characterization, process validation, and then ultimately through PPQ and launch with a team that's done it before. And I think that's incredibly important because we can talk about facilities, you can talk about scale and technology, but ultimately biologics manufacturing is the people. It's 100% the people, the team, the culture you build, and that experience inclusive of inspection readiness.
Yes, I wanted to also emphasize the point that Zandy made. So I'm Fraser Wright, a co-founder at Korea Therapeutics. So briefly, I've been mixed academic and industry background, faculty positions at Penn and Stanford, and also co-founder at Spark Therapeutics, and involved with 2 of the 6 licensed AV products in the United States to date. I did want to emphasize the point, the concept of platform manufacturing and establishing a manufacturing process that really can accommodate, to the degree possible, there's always some changes in the capsid, for example, if we're talking about AAV, for example. I think being able to perfect a specific process is very, very important, and of course, supports avoidance of changes later on in comparability studies. So.
Steve, I think you mentioned culture in manufacturing. Is that something you could say a little bit more about?
Sure. Yeah, I mean, you know, it goes to operational excellence. I mean, I'm very fortunate to lead a team of almost 200 colleagues who are operations-focused team. I think it's safety culture. I think it's operational effectiveness, right first time. And atop the list is really a service model to our customers, right? I mean, we are the manufacturer of record for our customers that entrust us with their lead or their second or third product. And you know, we take that responsibility very, very seriously. And so that's really an organizational mindset that we carry throughout the team.
Peter, I wanted to ask you your thoughts on build versus buy when it comes to gene therapy manufacturing. And I know Steve and Zandy will probably have something to say on this as well, but you know, what does this sort of mean? And what are sort of the key challenges around this decision making?
Yeah, so that was something that we undertook very early on, and we made a decision that we needed to have the strategic capability around manufacturing in-house. As I said, the foundation of the company is capsid engineering, but we can't get to patients in clinical trials, we can't get to commercialization unless we can manufacture at the right quality and yields. And so we made the investment in a 15,000 square foot facility on premises right next to our labs, And there's excellent collaboration, and to your point, culture of integration. And so one of the things that's really important is that we're bringing manufacturing in very early in the process. I think I mentioned even in the screening, from the billions of capsid variants down to that one, manufacturing screening is a very important one, and we won't move anything forward that is not at least as manufacturable as AAV9, the parental serotype. And so we made that decision. And then also, you were talking about consistency earlier, and having that consistency, we use HEC 293. It's also part of what we do in the capsid engineering process, and having that consistency all the way through is really important from going from the capsid engineering process to vector production to scaling up to 50 liter and 200 liter and eventually 1,000 liter. Having that consistency is super important. And also, in our minds, it also aids in the translatability using human cells in the manufacturing process for the ultimate, clinical trials and to help patients.
Yeah, Steve, would be interested to hear your thoughts on this, being the outsourced partner.
My current business is being a partner for innovator companies to be their development and manufacturing partner. I've had really diverse experience in my career on really almost all seats at the table, so I understand when boards make a decision one way or the other. I think the 10,000-foot view in my mind is really capturing capability and not capacity. So regardless if that's an in-source versus outsourced decision, what you should focus on is truly the capability, right? Because the focus on the facility, the engineering are important, but that's the commodity, right? The people, the process, and the process, guys, is the product eventually, right? So it's really, really important to focus on that capability. And when I talk to investors and customers about the decision, my focus is usually on cost of equity and the deployment of equity capital into all the vast priorities you have in drug development. And if you can find a partner that has the capability to scale and be able to produce at an early phase all the way to launching a product, I think you can deploy capital in other efficient ways within the drug development needs, right? And I think that's a choice most companies, and especially now in this financing environment, will have to take very seriously.
So I'll add in a little bit to that. So at Korea, we have made the investment, and it was quite expensive to build in-house CMC, I think, much as you mentioned as well. And I think that does really enable rapid iteration and improvements in your manufacturing process. But of course, it is capital intensive. But we found it's very important because we do a substantial screening process of candidate constructs. I think I heard this from Peter earlier. And we need to test them for manufacturability. And the fastest way, typically, we found to do it is with our in-house process. So we've made that strategic decision in-house at Korea.
And I'll give our answer as well. So I think we were a little bit early to have vendors that could do what we needed to do. I will say that one of the reasons we're in the position to have such a broad process and to be so well known by global regulatory agencies is because we had a pipeline. And that's very difficult if you're a CDMO where each customer has its own cell line potentially. So we were in a position where we have a deep pipeline of many different viral vectors and manufacturing, and we were able to build a process that today takes us very rapidly to GMP. Now, I said it takes 2 to 3 months. We now have 9 years of data and millions of data points on tens of different vectors, different capsids with different genomes in them. So we're able to use AI to help us narrow down the parameters that modify the process for every viral vector when we first put it into our cells, and probably in 6 weeks we've got it at GMP. So was it expensive? Is it expensive? Yes. We've got a 30-person process development team, but the time frame that we were as a company, would be impossible to be in the position we're in. J&J wouldn't be in the position they're in. with the potential to launch a product at the end of next year if we hadn't done it in-house. And we now have consolidated under one roof a huge amount of data around the process of manufacturing, which just isn't available out in the field because it's in one process. And that's extremely valuable, but yes, it's very expensive.
And I think what you'll find too is there's really an in-between too, like what's the best of both worlds for a new company starting today? Is it, do you want to invest in that development? Do you want that team in-house that can really refine the process before you tech transfer it for it anywhere to be manufactured, all the way to, you know, second source for commercial product? That's something too, where if your supply chain is flexible and agile, it probably has a couple sources of commercial product. And so I think as we'll see, as this continues to refine in the future, is it probably becomes somewhere in the middle of a hybrid on what the best thing for a new company or a mid-stage clinical company is.
If you were to look far ahead, maybe 10 years, what types of viral delivery do you think we will be utilizing? Will it be a lot of diseases or a few niche diseases? And if you had to name one thing as the biggest bottleneck that you foresee in getting from here to 10 years from now, what would you say? Maybe Fraser, start with you.
Yes, I can tackle that. So, well, first of all, I'm not quite sure, Liz, if we're talking about the manufacturing process or else the viral delivery type. Were you thinking of one or the other or both?
Whichever you think is more
Okay,
interesting.
well, I do have a strong opinion about manufacturing upstream processes. I mean, right now for AAV and lenti, which are the big two, I think, for gene therapy, viral vector gene therapy in any case at the moment, we are using transient systems. And they do really involve, for example, purchasing a lot of plasmid, GMP plasmid, for example, for transfection, or the creation of recombinant viruses such as baculovirus for the insect cell system. And I do believe that we're slowly but surely making very significant progress in constructing true producer cell lines that can be induced by small molecules. And I think that is the future. It is very complex with viral vectors because many of their components are cytostatic or cytotoxic. They inhibit the growth of cells. So we really have to overcome switching on, effectively switching on some of the key genes that are required at the time of vector production. So I think, a vision 10 years down the road is that we're using cell lines or producer cell lines as are used in monoclonal antibodies to produce viral vectors. So I'll leave that.
May I say something? Yeah. So I'm going to make a comment on both manufacturing and vision of the future because they're connected. So as I mentioned, we set up our company 9 years ago, and we had a vision of the future where we looked at genetic medicine and we went, what's needed to be able to apply DNA to diseases just like we apply RNA, small molecules, or biologics. What you really need is super precise control of the expression of whatever your DNA template is expressing. And we thought we'll build a company using today's technology, we'll have manufacturing, we'll optimize, but our aim is to be able to control any gene in any context with a small molecule of our choice. And so cut to 9 years later, we have amazingly invented a technology which is not transcriptional regulation, but it is a splicing-based mechanism whereby we can control mRNA production from a transgene using a small molecule where one small molecule produces one messenger RNA. So why is this important? It means that we use DNA now, not just to replace genes, but as the delivery device for any biologic therapeutic, any peptide, any antibody, any hormone. And we use it in producer cell lines. So imagine if you can literally control from 0 to any level you like, any gene product. That's what we can now do. So we can make a producer cell line, and with a different set of small molecules, which we design, it's not known small molecules, You can turn genes on and off at will. In life, what we can do is put a DNA template into the muscle, like one injection, give a pill, and natural GLP-1, GIP, glucagon is produced when you take the pill. Or you put that construct into a CAR T, give the pill, and the CAR is produced. So suddenly, we have a way of, in really tight timing, controlling any biologic antibodies. We can control every big company's largest antibody, from PCSK9 to Dupixent, all of them. And this, when we look again to 10 years' time, we've really provided a solution to dosing, right? The metabolic drugs actually should be short-acting. We can make them work way better at low doses. And for manufacturing, you no longer have to manufacture outside the body. Your body makes at exactly the right time the native, the natural, most efficacious form of your therapeutic. So in a sense, I see genetic medicines not just treating large indications, but now being a delivery device for some of the largest biologic therapeutics that are out there.
That's great. Very
It is,
exciting.
isn't it?
Yes.
The riboswitch is very, very exciting technology. I want to ask about cost, right? And I think, riboswitch is one example where gene therapy could actually reduce the cost of some of these expensive biologics. And we did get one question from the audience, thank you for that, on what's being done to help support the cost of manufacturing, the cost of manufacturing build out. The question is, what can the state, or I guess the local and broader government do to promote biomanufacturing that they aren't already doing to support your companies? Do you have any thoughts on that?
Yeah, I can maybe start there. Having built multiple businesses and biomanufacturing businesses in Massachusetts, I think there's the obvious piece of it, which you can incentivize the capital build. But to my point on people, is it's training, it's the pipeline of talent, it's continuing to invest in that. In a prior business with Mass Bio, we had just a great apprenticeship program, and we were able to bring talent into the company that could work in manufacturing after a 6-month apprenticeship. It was just a wonderful program. So it's, for me, the talent pool, the people, the ability to plug in there. And then once you start to build that and you have more and more biomanufacturing, talent pool's expanded, other companies can really pull from that as well at different levels of the company.
I'll say the same thing.
Yeah.
We built our second facility where there was a talent pool, and there'd been 20 years of the government really focusing on GMP manufacturing as a career, and it's a huge, this is in Shannon Island, huge benefit and huge difference. And we do the same thing, by the way, at universities. We have apprenticeships coming in and working with us. It's super important, because people are the most important aspect of GMP manufacturing. Lots of things, but that as well.
And I think this is a very important theme, and Korea has similarly identified, for example, Research Triangle is a great area with a high talent concentration and good support. I do want to say something more generally about cost of goods manufactured. I think that was part of our mandate here on this question. And it does go back a little bit, Xander, to the concept that some viral vectors right now require 10 to the 15th, and I won't even name that number. It's a quadrillion or a quintillion, I'm not sure. It's a big number in any case. And if you do work the efficiency of the genome, 2 or let's say 3 logs, that same amount will make 1,000 doses. So of course, this is an enormous opportunity to achieve more biologic effect with a smaller dose, I think is what it boils down to. At Korea, we are focused on direct-to-tissue administration. While most of, for example, the AV vectors that are used right now are systemically delivered, all but one, which is Luxturna, most of that material does go to the liver, and when the liver is the target, that's fine. For example, the hemophilias, but when the liver is not the target, most of the vector is actually wasted. It doesn't have a liver-specific promoter, so it won't express there, but it is actually essentially inert material. So the concept of delivering the gene in the tissue where it's required, I think is a very important concept going forward, kind of a visionary thing, I think, for gene therapy. We don't want to put it everywhere, or very, of course, very strong targeting mechanisms to get across the blood-brain barrier. So I think that's a real opportunity to bring the costs down. And a final point is efficiency of manufacturing. And I think we've talked about this already. You can have a log improvement in manufacturing yield just through good process development and optimization, perhaps more, and these are great opportunities to reduce the costs. Very important for prevalent diseases. For example, we're focused on this because at CREA we're focused on prevalent diseases.
And can I say one thing as well on that? You're absolutely right. So when we founded Mira, we only did local delivery because of the issues fraught with systemic delivery and tropism. Only local delivery, low dose. Manufacturing, I think we have improved yield 1 to 2 logs, but potency, that's 2 to 4 logs. So that's where your improvement in cost of goods is across the board. Local delivery, tiny doses, that's one of the reasons our Parkinson's drug is so cheap, because it's very, very local.
If I may, just from our perspective, I understand all the points that you were raising about direct delivery. Everything we do is IV, and the reason is for a few. One is because it's patient-centric. It may be cheaper to do manufacturing by doing direct delivery, but if you have a patient-centered focus, using IV, less invasive, particularly in brain disorders where you literally have to have brain surgery to do direct injection into the brain, carries significant risks to the patient, carries significant risk and burden to the healthcare system and cost to the system. So it's not just cost of manufacturing, cost to the healthcare system. And a lot of patients won't sign up for that. And so IV is really important, I think, for patient acceptability, but also we're using the blood flow. And by using the blood flow, you get more consistent tropism, more consistent penetration into all brain regions, as opposed to when you do direct injection, you're really only getting expression in the area where you're doing the injection and not that consistent brain-wide. And so I think, then it becomes our burden to figure out how to deal with the cost in order to create the most palatable, best patient experience. And that's where I do think the key enabler for us is the engineering of capsids. So we've essentially flipped the tropism that you would see from AAV9. AAV9, as was said before, almost all accumulates in the liver when administered IV, where we have significant tropism into the brain. We're getting to 70% of neurons transduce, and then very little expression upon scans or quantification in the liver. So I think things like engineering capsids can help address this issue. And at the end of the day, though, I think we have to have a patient-centered focus on what's going to be the best to deliver therapies to the people who are in need.
Just to be clear, we don't do local delivery for things that are systemic.
Yeah.
So muscle, salivary gland, and subthalamic nucleus, which is deep brain stimulation. So we would never treat a genetic disorder like that. We choose our indications because they work really, really well with local delivery. We don't try and treat things that aren't going to work with local delivery, okay, just to be clear.
Yeah.
And you're the same, I'm sure.
Yeah, we are too. But I would add, I agree that there's a great opportunity because there's really an infinity of possible capsid designs out there. It's a question of finding the ones, and it's a challenge to find the ones that really can achieve that specific, targeting at a reasonably low dose. Because I think the other thing we have to be aware of is that typically the systemic administrations right now are requiring high doses. So I think that's part of the equation as well. But
makes sense. Very interesting discussion. And maybe along those lines, we did get another question from the audience, which is talking about what's sort of the upper limit, right, of what's possible with gene therapy? Aren't there restrictions on what proteins can actually be delivered? They cited Zandy's example with riboswitch is exciting, but isn't AAV somewhat limited in terms of distribution or carry capacity, et cetera?
So the riboswitch works whether you knock it in, whether you deliver it with lenti, or whether you deliver it with AAV. So in the example I used of let's just say a triple gut peptide, we have a glucagon, GLP-1, GIP, a GLP-1, GIP, an amylin, glucagon, GIP. So we can very quickly, just like Moderna could within weeks actually, make a message for the right COVID sequence, we can, within a couple of months, make a construct that can be delivered into the muscle, for example, and when you take a pill, those gut peptides are released. Now, alternatively, in other systems, in cell systems, and whether that's cells that you're encapsulating and transplanting, or in CAR T cells, we actually knock in our cassette, right? Or we deliver it with lenti. So the riboswitch, the control technology, is not limited by delivery. It is itself a delivery of the biologic, of the gene therapy product. So we've got in vivo data representing every one of those examples in tens of different types of genes. It can be used anywhere that a small molecule can get to. Go ahead.
And yes, and I'm keen to weigh in on this question too. So I think it is true. It's well recognized that AAV has a packaging capacity of around 4.7, maybe 5 kilobases, 5,000 bases. But we should also recognize that the latest, I believe it's the latest AAV product licensed in the US is Elevidys. You know, it is for Duchenne's muscular dystrophy. In fact, dystrophin, wild-type dystrophin, is the largest gene in the human genome. And of course, there was a lot of engineering that went into making a microdystrophin that would fit within an AAV vector. So, you know, there are ways around the space limitation, but it is a real limitation. I think it's a challenge for factor VIII, for example, for hemophilia. But I would also say that, for example, at CREA, we are interested, one of our themes and one of our programs is vectorized monoclonal antibodies. And a monoclonal antibody, an IgG, heavy chain and light chain, that will make an intact monoclonal antibody will fit within AAV. And I think we all recognize the enormity of the number of possibilities there are out there using vectorized monoclonal antibodies. Anything that you'd like long-term expressed could be done by the gene therapy approach. So that's, you know, I think there's a large range of opportunities, even given the restrictions of the packaging limit. And of course, lenti's larger, and there is on the horizon the use potentially of recombinant herpes simplex virus for durable gene delivery as well. And of course, it has an enormous packaging capacity, and of course then non-vector approaches, and I think, yeah, as Andrea talked some. So.
Yeah. We've been hearing a lot all day about strategic partnerships from every possible angle, so I wonder if we could just talk about what is the single most important partnership that you think you have established so far. Maybe Steve, is that something that you'd like to
Sure. Yeah.
comment on?
You know, a little different on the CDMO side because you typically aren't investing a ton of R&D dollars into your own platforms. And so the way we access some of this technology is via partnerships. So when we talk about a producer cell line, for instance, access to that so we can provide it to our customers is through a partnership with CSL. And so we tend to focus that way on things with producer cell lines, with synthetic plasmid, and sort of how do you impact yield, and then how do you look at downstream processes and improvements agnostic of the upstream process. And so that's where how we focus on partnerships as a CDMO.
Peter, do you have a quick answer to that?
Yeah, I wouldn't dare differentiate between our 4 partners and say one is more important than the other. But I will say partnership has been really critical to us. We have partnerships with AbbVie, both in CNS and in ophthalmology, with Lilly in CNS, and we also have a small manufacturing partnership where we're manufacturing for a sister company in the Westlake and Versant portfolio. All of them have been really enabling for us in various different ways. But I mentioned earlier our CRISPR Therapeutics partnership as well. I think having access to world-class capabilities, having access to different cargoes has really, and just, you know, learning and working together, and us providing a service and helping our partners, but then us learning from our partners as well, all 4 of those partners has really been enabling for us.
We had a partnership with Johnson & Johnson, and they paid 100% of manufacturing and clinical development. and they owed us a 20% top line. That partnership allowed us to build the manufacturing we have today and a lot of our capabilities. And really importantly, pharma companies focus on BLA and commercialization, not from a manufacturing perspective, getting things into the clinic. And that was transformative for our manufacturing in this company. We learned, we understood what the target was, and we also had global regulatory interactions across the board in manufacturing. So that was super, super important. Johnson & Johnson then bought back the lead program, and we, Mira, entered into a commercial supply agreement with them. So a complete change in economics and structure of partnership, but that becomes validating for our manufacturing. But that's definitely been the most important partnership from a financial, a learning, and ultimately as we move forward in a financial way.
Fraser, last thoughts on the partnership?
Yes, okay, thank you. I realize we're just a little bit over time, but I did want to, I'm going to take an advantage here and say what I think is a very critical partnership. I agree with these important partnerships I've discussed. But internal partnerships, I think within a company, I know at Korea we've made a real effort to make sure that we have strong fluidity between our basic, our applied, our process development, and the manufacturing right across that board. I think that's a very important partnership. And I'll just use that to illustrate one point, the importance of quality of and characterization of, for example, viral vector products that are made. It's such a key component. I think we're emerging to a full understanding of the critical quality attributes of, for example, viral vectors. And it's very important to make sure that we have the perspectives right from the basic research all the way to the GMP manufacturing, all of those aspects to make sure that we can accelerate and optimize the highest quality. It's a major focus for us at Korea, the internal, as well as, of course, these important external partnerships.
Fantastic. Well, with that, I think we're at time, so please join me in thanking our panelists for the engaging discussion. Thank you.